Hello students, welcome to EPG Pathshala. I am Indira Ghosh from JNU. Today we are going to talk on the module structure based designing or docking and scoring under the paper computational biology. Our learning objective is how we identify new compounds using structure, also the algorithm of docking compounds at the active sites, and the disadvantage and advantage of each of these methods as it has been going through the applications. We would like to discuss in the beginning what is the challenges of doing structure based design. The most important challenges are we are handling a large macromolecule and it finding its active sites, which is really a time consuming work, and that requires CPU time as well as it requires large amount of hard disks. The reason is both the cases the time requires not only for more compounds to store and do the high throughput screening, it also requires the accuracy of data to find out how the best way we have put the active site and the molecule and tested the molecule at the active site. This is the most difficult challenges in the structure based drug designing. However, solutions are that instead of searching for any compounds, we do search for a focus library. This is one. And doing the designing, before doing the designing, we can do a virtual screening of diverse library. Hence, designing compounds to start with a different scaffold would provide us very good starting points. Also, we can combine the structure-based as well as ligand-based to evolve a better strategy for such challenge. Our target for doing such docking experiments are quite different ones. One is enzymes. Major case studies have been done with enzymes. Then it can be enzymes or protein and DNA, and it could be even just DNA molecules. Also carbohydrates. Now, it may not limited to only enzyme, DNA or carbohydrates. It can be protein-protein interaction, or maybe to stop the pathway. That means many protein can be inhibited in the same pathway. Also, the target systems can be cellular metabolism, which includes many complex molecules in together. As I have described earlier, that enzymes are one of the most important target for proteins, structures, etc. from protein data bank. There are different kind of groups and we would like to show you that first. One is they have this catalytic triad or dyad or whatever, like aspartic acid, histidine, serine. Plenty of these amino acids plays the most important role in the active sites. Nonetheless, also the active sites are designed by the proteins in a very different way. And if we look at the structure point of view, here are two of them. Both are, one is bacterial serine protease, another is mammalian serine protease. However, their structural folds are little different. In one case, it is only the beta structures. Another case, it has a beta alpha structure. However, both of their active site triads are aspartic, histidine and serine. All the same, almost the same. Similar thing can be also observed on a other level, and that is shown here. Similar structure, almost identical fold structure of protein. Here it's shown called Tim-Barrel proteins, but biologically or functionally, biochemically, they are different. One has tripose phosphate, triose phosphate isomerase, another is resin. These two are found to be very interesting targets for also for designing because they are very important. Here I show you not only their different proteins have different folds or similar fold having different biochemical reactions, they might have flexibility. Not only they will have flexibility, they have flexibility related to function. Here are 2-3 examples of it. One example is right on the top, which is binding to substrate. This particular protein, which is E. coli aspartate aminotransferase, actually changes its cleft, and it between the two domains, here is the green and here is the yellow, you can see these two, and this cleft between them narrows, and that's why the catalytic residues come near to each other. This is very much important for functions, hence open structure and closed structures both is being shown here. Another example, which is even more important, and especially tRNA binding protein. This is a important structural protein, and designing is against this to have a drug is very important. Here, it not the substrate binding. Substrate is actually a tRNA, which is shown in gray colored small beads. The yellow color, that is the GTP binding, brings these two domains nearest to each other so that actually the substrate can bind. So here is another example. Earlier example was catalysis can be done. Here the example substrate can bind. This kind of flexibility related to the functions makes it very difficult to use a static model of structure as your interaction evaluation. Most important well-known drug is HIV protease, where two units together forms the active site. But more than that, when the inhibitor binds inside, it actually closes the structure. And so too happens when any kind of compound or substrate it binds, it closes the structure. This is the interesting. Because of this, the important haloperidol, which is an interesting compound found to be against HIV protease, was found, actually been designed to close the two flexible flaps, which is shown in blue. However, the most important part why we are able to accept the proteins and design them as a mechanism being different, structure being different, flexibility is there, is shown in this particular article as well as this plot. Taken care of all known protein structures, when one looks for several structures of acid protease of different systems, one found that the catalytic diode, which is shown in B, especially shown in B, all the catalytic diode aspartates, they superpose on almost on each other. And that is a proof of that when anybody is designing an inhibitor, a class of proteins generally will bind to the same inhibitor almost all the time. And this raises the question of selectivity to bring into the picture of when we design substrate-based molecules. As I have narrated earlier, that there are reasons for looking forward to finding the new ligand designing using the active site. However, what are the ingredients where we should start with? It must be related to the interaction of active site with the ligand what we are going to design. And these interactions actually are physicochemical. They can be highly specific, like it should be directional also, like hydrogen bonds. It has to have a quality called non-covalent interaction. Most of the biological interaction are done or for stopping the function or having the functions are designed by these non-covalent interactions like hydrogen bonds, like hydrophobic interaction, etc. However, some of the times one needs to interrupt the interactions which are covalent bonds. Now, it has to be working under the presence of water because all the cellular function happens in water. However, sometimes to exclude water, reaction might happen in membrane. Now, this interaction should be kinetics and thermodynamically evaluated so that the bindings, energies, etc. can have these properties. Also, one must understand the ionic environment of every interactions do change depending on which organism and which organelles the system is actually interrupted. As I have discussed earlier, The major factors have been depicted in the previous slide to consider when we design the structure-based designing method and virtual screening. Here I would talk about what are the methods, what should be the methods, and based on that we will discuss about different algorithms. Now, question is, the docking protocol will have a search strategy, that is, in the active site we should find it where the small molecule can bind to. That means what is the complementary interaction it has. Another is, once it bound to different complementary interactions, maybe there are n number of interactions and the small molecule binds with x number of interactions. Then probability is that it can bind any n because n is always greater than x. So we must evaluate, and that's we call it a scoring function, which interaction is strong. That means out of several possibilities of n combinatorially, how many of them this x complementarity is emerged and it is better interactive than others. So we must not only have a strategy for docking, we should have a strategy for scoring as well. Such algorithm should make optimum number of different conformer of the molecules, also include as much possible structure which is experimentally known. A rigorous search must be done, which is possible to binding mode of all the 6 binding mode of the molecules. Now, what is this degree of freedoms and how many it should actually account for? That is next slide. Now, search algorithm has the most difficult part, because a ligand with 4 rotatable bond and 6 rigid body alignment parameters, you can have a space which can be roughly calculated with the positioning in a cubic site would be 10 to the power 3 if your active sizes. And if we change the angles by 10 increment and translation parameter by 5.5 grid, we land up rigid body 4 into 10 to the power 8. Then 6 into 10 to the power 14 configuration of the molecules. Together, you require, if we have 10 configuration per second on any computer gives you the strategy of searching and scoring, you require 10 to the power 6 years to do these calculations. Hence, we must find a strategy, better strategy than just doing it as brute force. As I talked to you that earlier, that the search algorithm has to be so efficient that it should be running a huge amount of calculation within seconds. But still, as you know, it is impossible if we do not restrict our search area. Also, we must understand, for our modeling study or structural knowledge of active sites, protein and small molecule, we have understood there are something called preferred or favorable space of interactions. So, one can, to estimate the interactions, energies, free energies, etc., one can calculate only the favorable space. One can avoid the diffusion mode. That means one can put the molecule right in the active site instead of letting the molecule enter as it normally does. Also, we can look into the ligand's allowed conformational step, as well as we can consider the receptor not to be flexible. These few opportunities are explored in the next few techniques which have been described as a docking techniques. I talk now about the scoring function. What should be the scoring function? Actually, scoring function can be very exhaustive, which is actually finds all the force fields which I have discussed earlier, and interaction energies and free energies, etc. Or it can be a knowledge-based function. Now, 2 types of scoring functions are normally used in this kind of research in docking. One is full scoring function, where actually finally after the algorithm finds out most suitable molecules to bind and most suitable pose or orientation, one uses very much thorough calculation of scoring functions. However, when one evaluates the poses, that is how many times and wherever the molecule should be fitting, one uses rather reduced functions because the time limit, because quickly we have to throw away molecules which are not going to be best in the active sites. There are few assumptions which also causes the limitation. One is, we actually have considered that is a energy hyperspace. So we actually never consider some of these large terms like electrostatic calculations. It's a huge interactions. Most often we consider only hydrogen bond interaction, and we actually eliminate whenever there is a high energy surface. So one side we use this steric hindrance to reduce our conformational space searching, interaction space searching, and we use favorably our hydrogen bond energy most of the cases where the interaction energies are calculated. Now what it happens, either it neglects solvent effect or it uses a limited solvent effect. This is where comes the limitation because of quick calculations. Another is we totally neglect the solvent and ion which always being bound at active site. Several cases they do affect, as I have mentioned earlier mechanisms, earlier my modules has some of the nucleophiles are water molecules, and we lose those water molecules while doing this quick docking and searching. But more than that, actually many interaction, as I have told earlier, are possible in the active site. Actually we do not evaluate all of them. Hence a small molecule only have a chances maybe 5, maybe 7, maybe 10 interactions. However, the complementarity demands that it can have any combination out of 20 or 30 interaction possible. We do not put any penalty for this loss of interaction or which should have happened. These few points to be remembered when we evaluate or rank our molecules proper phase. Also, when we calculate scoring functions, it will come back to us and haunt us basically to find the proper poses. Now I'm going to describe you most of these principal docking methods, that is majorly used docking methods. Now many techniques and names of the softwares are written. However, basically they belong to one or two, at least 5 different category which has been shown on the right hand side. But 2 other methods I haven't described here are Monte Carlo and molecular dynamics methods, which are pretty expensive and can be handled differently later on. However, on the right hand side, I have actually numbered them by top to bottom in a reverse way. The reason is it depicts the computer time requirement for the lowest one, that is A, is a huge done. However, the accuracy is a very different question. But the time required for doing the topmost, like genetic algorithm techniques, are much faster than the time required by taboo searches. So there are 5 major methods I would describe later. One is taboo search method, which I will explain many times on many methods available and the softwares available. Distance geometry is another. Fragment based methods, which are very popularly used in doc and FlexX, are also will be described. I will not describe too much detail of this point complementary method, which actually is a very time consuming method and not so used in docking methods. Genetic algorithm is the most recent phenomena and has been utilized, and most popular software called Autodoc based on the genetic algorithm. I will go through quick now which depicts description each one of them, which I have described earlier slide. This is a method which is systematic rigid body search, and it has been applied. Force fields are applied very well. However, test in the pudding is only eating it. Hence, the green color slab shows you that how many one screening, virtual screening, has yielded how many hits by this particular method, and among them how many are really went for good inhibitors. So every slide of this will contain this evaluation also at the bottom. There is another which is called taboo search, which is a technique or algorithm follows that it calculates the poses and it retains the poses. If it gets a new pose which is actually not found or better than the earlier, it keeps it, otherwise it throws away. So by this method, it can find very many compounds. This is a distance geometry based method, which is hydrogen bonding, etc., and takes care direct flexible of the ligand binding. The structure of this code, either by PMF or one type of scoring or PLP, these two are actually empirical scoring, not so much of energy level scoring. This is called fragment-based method. Fragment-based method basically divides the ligand into separate positions, very many portions, and then what it does is it goes on docking them in different part of the protein active site. Now, the difficulties are that it assumes as if the energy surface is a very continuous surface. Also, it has a difficulty at the final level, that is how we decide which functional groups are important more than others. This is, one can utilize this pharmacophore, which is taught in ligand-based designing, and use this technique to identify the complementarity of the active site, which has been utilized also in some of the following methods. FlexX is one of the methods, is the method which actually FlexX takes care of this, many of the favorable interaction in a pharmacophore mode. So it brings the elements of the ligand-based design into the active site design, and it uses scoring function as Bohm's function, which is also an empirical function which actually just calculates the interaction of hydrogen bonds, etc. This is details about the steps and talks about the validation, which is important to note here. Here, the 19 different complex from crystal structure were done and 10 were successfully reproduced, however, with a very minimum RMSD, which is at 1 angstrom. Now, that is sufficient, good to prove that almost 10 were successfully done, because 1 angstrom RMSD can be differences between the 2-3 structures, which are two different structures. However, this is the very advancement. You can use water sometimes. We can use even flexibility of the receptor in this method. DOC, on the other hand, is much more accurate and not so fast technique. However, uses the shape as well as complementarity, both taken care of, and at least it makes a larger sphere in the active sites and tries to fill it up of the active site. At least 4 pairs must be found uniquely, so that the stereochemical geometry are retained in the ligand and receptor binding. As continuation of DOC principles, scoring for this best DOC ligands is done by mapping the ligand onto the contact grid, as I've described earlier, and this force field is used, exclusive force field is used, like amber force field mostly used in DOC, which gives a scoring function as if it is more like a free energy of binding. The best scored ligand in the databases are actually could be find and rank them according to their binding efficiencies. This reference is fully given in this particular shown in 1997, and many others followed up. DOC fragment-based method also uses more or less space orientation as well as fragment-based. DOC uses bipartite graph. This graph method, in joining with click method, the finding the multiple clicks, orients the spherical DOC fragments appropriately in the active site. The protocol is anchor the fragments, then backtrack all of them, then minimize by amber. DOC also takes a very strong energy force for scoring. Now, 10 structures from complex were taken and 7 were DOC reproduced almost the crystal structure within the 1.03, but rest of them also were reproduced almost less than 2 angstrom, which actually talks very high and good about DOC. But best part of DOC is, which is been done few years now, is takes care of flexibility. Also, it can evaluate the scoring functions using a continuum solvation model, which makes DOC to be more appropriate for taking active site, special active sites where pH dependency or ion dependency makes very important biochemical reactions. Ludi is another designing which also kind of fragment designing, and it may make a lot of fragments. Actually what it does very good is, if you have a scaffold identified already and want to look for selectivity, Ludi becomes very de novo design protocol. What it does is, it takes the scaffold and puts in the docking site or puts it in proper algorithm in the active site. Then you can substitute every positions with different kind of, which is comes from ACD database or a small substituent molecules orientations, and it just goes on substituting every place and finds the best interaction designing is done by Ludi. So I would like to go to next slide, which talks how the steps of working out with Ludi. Fit the ligand's active site, then do the fragmentation library and use it. Prediction has been done with HIV protease inhibitors and DHFR inhibitors, and 2 of these fragments were predicted and experimentally found to be improved in the binding. Even we have done some of this use of Ludi to do the selectivity, very efficient method to do the selectivity and lead optimization phase. Here I show you the example which we worked on to do the selectivity. In the bottom it shows, and basically it does the interaction site mapping. So there are geometric principles, there are grid principles, and there are contact distribution with the small molecules. All the 3 are utilized with this Ludi method, and then it joins the fragment as a chemist knowledge used and builds the whole molecule. Alignment of fragment based on hydrogen bond motif. This is a very interesting proposition, and actually it worked, but well, Ludi would be better off than this because Ludi takes care of scaffold locking also. However, that method was developed to do very fast calculation, as it has been found the hydrogen bond is the most important interaction for specificity in the active site and the interactions to be inhibited to be designed. Hence, a mapping or motifs of hydrogen bond search was done, and then choose the fragments to put in dummy atom method. But has the difficulty is, it has been applied to DHFR and ribonuclease T1 and very efficiently found the inhibitors. However, the difficulty is, if the molecular fragment is large, Adam finds it difficult for only using hydrogen bond. The reason is space filling or complementarity in shape is not included in this method. However, DOC does much better than this method. Hammerhead is another method which is also fragment based. Discusses hammerhead over here is the method of hammerhead where it's like fixing the head of the hammer depending on the interaction. So it is more like the method of Ludi. However, it does not retain, it actually starts with a large head fragment and does not connect the chemistry. It does not work on the chemistry, but it just provides you the small fragments on the active site interactive method. 4 complexes were reproduced by around 1.7 RMSD, and this has been used for streptavidin and biotin ligand. Actually, Hammerhead does very good once you know the large space filling molecule already, like in the case of streptavidin and biotin complex. So, one can use protein docking probably, and a better method of this has come up now for Hammerhead, from Hammerhead, which is very well-known molecules, and slide earlier version, then glide. These are mean field theory. So it does reset every ligand, overlaps all the geometries, and goes on finding as a dynamic way the methods. So these all methods are basically fragment based, started with fragment based, but they do anchor one of the points, at least 3 matching point. So normally they take hydrogen bond donor, acceptor, and hydrophobic ring centers as their first three sites, and then start performing search for the molecules in every place as possible. What could be the designing? Apply to database screening, and even with water it has identified almost 50% true hits. And Glide actually is a very interesting package which has come up and become a part of a very good program called Schrodinger software package, where you can use the energy functions, actual energy functions like amber force field or charge, to do the ranking or to do the orientation fitting in the while doing the docking. I have discussed already three major methods, which is shown here: taboo search, distance geometry, and fragment based docking method. Elaborated their advantage and disadvantages. The next topics would be to go for genetic algorithm in detail and how that method has been developed and what are the methods there. So I have described the basic principle of docking, type of scoring done during docking, as well three different methods which I have described, elaborated, their efficiency in docking and applications. And the next slides I will follow up is the method which is most popularly used, called autodock, and principle behind it is GA or genetic algorithm techniques.